Hello children, welcome to my video class. As per your requirement and need, we are step by step progressing towards the sexual reproductions in flowering plants, the different structures and events. In the previous classes, we have discussed to you about the microsporangium, microspore mother cell and microsporogenesis, formation of male gamete. We have discussed about megasporangium, the structure. We have discussed about the process of megasporogenesis, how the female and male gametes are produced. Now, if we progress further, the process of ultimate aim, wherefrom we can get the next generation, that is the event, that is fertilization. But before fertilization, the male gamete has to approach and reach to female gamete. That is, the pollen grain has to land over the stigma of a pistil. Now, let us discuss in detail the process of landing of pollen grain to the stigma of a pistil is called pollination. Now, let us discuss in detail what are the modalities of pollination. What are the different modes by means of which the process of pollination can take place? If we talk about pollination, see, in the case of flowers where both the male and female reproductive part present in the same flower, there is a chance that the pollen grain from that flower can transfer to the stigma of the same flower, that is stigma of a pistil present in the same flower. If it is the case, we use a term called autogamy. What is it called? Autogamy. Where the pollen grain transferred to the stigma of the same flower. That means if I draw a simple diagram here, suppose this is the stigma of a pistil, and here is the anther. The pollen grain gets matured, present in the same flower, the pistil and the stamen. This is the external part, the petal, the sepal. Now, if the pollen grain once released and transferred to the stigma of the same flower, then this type of pollen transfer we call autogamy. But in other cases where the male flower and female flower both are present, but the pollen grain, instead of transferring to the stigma of the same flower, is transferred to the, suppose this is a bunch where another flower is there, say suppose this is another flower, having a stigma style and the pollen grain, instead of transferring from this flower, this here from the stamen to pistil, instead of transferring from here, if the pollen grain from this flower transferred to the stigma of another flower of the same plant, then this is called as, so one we have discussed about orthogamy in previous case, and here we use a terminology called geitonogamy. So what is geitonogamy? Geitonogamy is the process of pollen transfer in which the pollen grain from the anther of one flower is transferred to the stigma of some other flower of the same plant. So let me write so that it will be very clear to you. In this process, the pollen grain from anther of one flower is transferred to the stigma of other flower of same plant. So in first case, orthogamy, we have seen the pollen grain from the anther of one flower is transferred to the stigma of the same flower. This is the first case, orthogamy. And in second case, in the same plant, we are having numerous flowers wherein the pollen grain from one flower is transferred to the stigma of other flower of the same plant. This is what we call geitonogamy. So can you tell me whether it is orthogamy or it is cross-pollination? Whether you can consider it as cross-pollination or self-pollination. Genetically, it is a self-pollination because there is no exchange of genetic material not coming from some other source of some other plant of the same species or of different species. A third type of pollination we shall discuss now, that is third type. Shall I rub this part? A third type. What is that third type? That is called number 3, xenogamy. So we have talked to you about geitonogamy, we have talked to you about self-pollination, that is orthogamy, and we will be talking to you about xenogamy. Xenogamy. What is xenogamy? So geitonogamy is very clear to you. Orthogamy is very clear to you. Now xenogamy. What is it? It is the process in which the pollen grain from one flower of a plant is transferred to the stigma of other flower of other plant. So if I make a sketch of this phenomenon, say for example this is one plant having a flower, this is another plant having a flower containing female part, other parts I am not drawing. So if the pollen grain from this flower is transferred to the stigma of other flower of other plant, this is completely new plant, different plant of same species or some other species, then this is called as xenogamy. So xenogamy is a perfect cross-pollination. So genetically, autogamy and zetynogamy is a kind of self-pollination. Although zetynogamy appears to be a cross-pollination, but no, genetically it is a self-pollination because the pollen transfer takes place within the same plant, the flower present, the different flower present in the same plant. So there is no genetic exchange of material. But here in xenogamy, it's a perfectly cross-pollination. Is that clear to you? Now, question is, what are the agencies which will transfer the pollen grain? Now, can you just think about a perfect autogamy? Can there be a perfect autogamy? See, in this case we are talking about autogamy, but it may not happen so. Why it may not happen so? Suppose by that time the pollen grain is supposed to mature from this same flower and land over stigma. Before that, the pollen grain of some other flower, of some other plant might transfer to this stigma. So there is a chance of cross-pollination. In most of the cases, it is the case where there is every chance for cross-pollination and cross-pollination takes place. So self-pollination or autogamy is a chance factor. It might take place, it might not happen. But if you want to ensure this self-pollination, that is autogamy, there is a case. Say for example, in the case of common pansy plant, Viola, oxalis, these are the examples. So Viola plant, if you take, it bears two types of flower. It bears two types of flower. One common flower which is normally exposed just like a other flower, having petals, the female part and the sepals, just like normal flower. But they are having another type of flower where the petals are always closed. As a result of that, so this flower, which is just common like any other flower, where on course of maturity the flower opens and the stigma and the anther are exposed. So exposed flower. This type of flower we call chesmogamous flower. What is this type of flower we call? Where the flower is exposed on case of maturity, the different parts, the stigma, that is pistil and the stamen are well exposed. So this type of flower is called chesmogamous flower. Chesmogamous flower. So in case of chesmogamous flower, there is a chance for cross pollination or by chance self pollination might take place, but it cannot be ensured that self pollination will take place. There is every chance that cross pollination will take place, just like the example I have given. But the other flower which always remain closed, never expose the flower which the petals are always covered. The sepals are covering, sepals and petals are covering the stamen and stigma. So the stamen and stigma both are there. That is the pistil and the stamen both are there. That is the male and female part, reproductive part, both are present in the same flower. But the petals are always covering the stamen and stigma. The sepals are covering, the petals are covering. So, the flower, the male and the female part, the stamen and the pistil doesn't get a chance to open up. Hence, they are not getting a chance to open up. They are always closed. Within that, it is closed. As a result, from outside source, the pollen grain doesn't get a chance to make an entry and pollinate the flower. So whatever the pollination takes place within the same from the stamen, the anther which contain pollen grain on maturity, it will land over the stigma of the same flower. This flower is called clistogamous flower. What is the flower called? Clistogamous flower. And in this clistogamous flower, there will be always autogamy. Is that clear to you? So, in terms of modalities of pollen transfer, it can be autogamy, it can be zetonogamy, it can be xenogamy. Xenogamy is a perfect cross pollination. If you want to ensure the autogamy, rare chance. But as I told you, this is a flower that is clistogamous flower where there will be always autogamy. Is that clear to you? Now, coming to the point, agencies of pollen transfer. The agencies for cross pollination, you require an agent who will transfer the pollen grain from anther to the stigma of a pistil. The agency of pollination can be of two types: abiotic agent and biotic agent. Generally, abiotic agent is very less number of cases, especially some monocots, water hyacinths, or such types of algae, bryophytes, and even certain pteridophytes. There is a chance for pollination through abiotic agent. Out of the abiotic agent, air is the most common abiotic agent. Other than air, water is also. But as far as abiotic agent is concerned, it is mostly the air which are acting as an agent for pollination, that is medium through which the pollen grain transfer. If the pollen grain are to be transferred through abiotic agent air, through air medium, then there should be certain characteristics of the pollen grain. What could be the characteristics? the pollen grain is supposed to be very, very light, so that they can be carried by means of air to certain distance. Number one, the pollen grain should be light. If it wants to transfer by air, it should be light, very light, and it should be non-sticky. Number two, non-sticky. So that it can easily be carried. And for the female part, it has also certain characteristics. That means the female part should be feathery. That is, stigma should be feathery, so that it can easily attach and give a landing space to the pollen grain which are carried by the air. Is that clear to you? If you see the structure of a corncove, which is nothing but a inflorescence, compact, so many individual flowers are attached with it. So many individual flowers are attached with it. Individual flowers are attached with it. Each flower is having ovary with single ovule. This is the structure of a corncove. The male and female parts both are here. This numerous flowers are here, forming a structure called inflorescence. Now, here within this structure of inflorescence, combination of so many flowers, bisexual, both male and female parts are there. Male parts are generally well exposed, like this, the anther, these are the anther, well exposed. And the female flowers, especially the stigma part, they are collectively form a bunch of here like what you call as tassel, which is giving a landing space for the pollen grain when it is released. Is that very clear to you? In case of pollination done by air. In case of water pollinated plant, generally very less number of cases are there where pollen transfer is taking place through water. But wherever it is taking place, it is the water current which are carrying the pollen grain and reaching to the stigma. See, certain plants like Vallisneria, like Hydrilla, aquatic plant, where we see these types of pollen transfer through water. But in this case, if the pollen grain are carried by water and has to remain for some time in the water, there is every chance that pollen grain can get damaged in water. So in order to prevent that pollen grain, and the pollen grain here is almost ribbon in shape, and they are covered by a mucilage coat. This coat prevents the pollen grain against water decay, so that they can get a chance to reach to the stigma. So this is about the abiotic agent. Now coming to the point of biotic agent. This is the abiotic agent. Abiotic agent we have discussed. Now coming to the biotic agent. Biotic agents are numerous. It can be insect like bee, like honeybee, like bird, or like other animals. The most common pollinating agent is the honeybee or bee, which are in search of nectar, moving from one flower to other flower, and while moving their antennas are stick to the pollen grain, and when they are moving to some other flower, just the pollen grain from one flower is transferred to the stigma of the other flower, and the process of pollination takes place. In certain cases, we are having a very good example. Example like certain moths and flower, certain example of flowers are there where moth and flower are having a coexistence. They are both benefited by each other. How? The moth is visiting the flower because of nectar or honey, and the moth, while visiting the flower for nectar or honey, the pollen grains stick and transfer and pollination takes place. And the moth is not only been benefited for nectar, but they deposit their egg within the locule of the ovarian locule inside, so that their egg can be protected well. And when the ovary by the time converts into fruits, seeds are developed, and within that seeds, the egg by the time gets matured, hatched, and larvae coming out. So both the way they are benefited. So I have given you a brief idea about the different modalities of pollination. Cross-pollination, that is self-pollination, xenogamy, cross-pollination, and zetynogamy, that is a pseudo cross-pollination but genetically not a cross-pollination, and autogamy, and the agencies of pollen transfer. Now, once the pollen is transferred, then what are the events takes place? We shall discuss now. Be very, very careful, observe carefully, so that you can get a very, very clear idea. Once the pollen is landed over the stigma, that is, the pollination gets completed. Pollination gets completed. But here are so many hazards. What is it? The timing of pollen grain maturation and maturation of pistil may not be synchronized. That means timing. If both are not so synchronized, then the pollen grain will fail to germinate over the stigma. So they have to mature at the same time, otherwise we cannot get the process of pollination to be completed. Number one. Number two, so number one, synchronization. With respect of maturation of pollen grain and pistil. Number two, if the pollen grain and pistil are matured at the same time, even then the fertilization cannot take place. That means it will not proceed further. Why? Because the pollen grain may not be a compatible one. Compatibility. What is compatibility? Means right type of pollen grain. The stigma, or as a whole the pistil, has the ability to recognize whether the right type of pollen grain is landed over it or not. If it is not a compatible one, then further germination of pollen grain will not take place. Germination of pollen grain and further proceeding of fertilization and post-fertilization event only will take place if there is a synchronization of timing with respect to pollen maturation and pistil maturation, and at the same time the most important is if the right type of pollen grain is landed, right type. So how it will be determined whether it is the right type of pollen grain or wrong type of pollen grain? There is always a continuous dialogue called chemical conversation. What is it? Chemical conversation. Chemical conversation. Chemical conversation. Between pollen grain and the pistil. This is what we call pollen pistil interaction. Pollen pistil interaction. Otherwise called pollen pistil dialogue. This dialogue or conversation between pollen and pistil is mediated through chemicals, and that is what we call chemical conversation. By virtue of that conversation, if the pistil feel, understand, recognize that the pollen grain so landed is a right type, compatible type of pollen grain, then it will allow it to germinate. Now what happens if the germination takes place? That means if the pollen grain is compatible one, then pollen grain, the XII covering through the sporopollunin, the in time will come out in the form of a small tube, what we call as pollen tube. Pollen tube. Now, within the ovary, you are having ovule. We are having ovule. Covered by integument, say outer integument and inner integument, covered by integument. In previous class we have already discussed the egg synergids, two synergies, and center there is egg, the polar nuclei, the antipodal cell. Now, once by virtue of pollen pistil interaction, the pistil recognizes that the right type or compatible type of pollen grain has landed over, it will allow to germinate by forming a small tubular structure called pollen tube. By that time, the pollen grain is at two cell stage, the vegetative and the generative cell. Now the pollen tube, by that time the pollen tube so formed, the generative cell divides to form two male gamete, which reaches at the tip of the pollen tube. So let me draw further extended pollen tube. This pollen tube is passing through the stylar tissue. These are the two male gamete formed within the coming within the tip of the pollen tube. Now let me draw further extended pollen tube. This pollen tube further increasing its length, entering the ovarian locule, and trying to make an entry, just concentrate here, trying to make an entry through the micropyle of the ovule where there is no integument covering these two male gametes. Now once it is approaching the micropyle, the filiform apparatus present at the tip of the synergids, through which the pollen tube make an entry into the ovule, and once enter to the ovule through the filiform apparatus of the synergids, they release the male gamete, one of the male gamete. So let me draw it here separately. Only the ovule having some big size I am drawing here now. These are the synergids, two synergids, and this is the egg, the polar nuclei. This is the antipodal cell at the chalaza end. Now, one of the male gamete fuses with the egg. This is the first male gamete. First male gamete fuses with the egg. And the other male gamete, so two male gamete, other male gamete, the other male gamete fuses with the polar nuclei. So one of the male gamete fuses with the egg and the other male gamete fuses with the polar nuclei. When one of the male gamete fuses with the egg, this process we call syngamy, and the resultant product of syngamy is a formation of zygote. Let me write here. Once one of the male gamete fuses with the egg, the process we call syngamy, and the product is zygote. And the other male gamete, second male gamete, fuses with the second male gamete, the second male gamete fuses with the polar nuclei. This is the polar nuclei, and another male gamete comes here. The resultant product is primary endosperm cell, or primary endosperm simply called, and this is called, so this phenomenon is called triple fusion. Triple fusion. Why it is called triple fusion? Because already two nuclei are there in the polar nuclei, and third one coming carried by one male gamete. So there are resultant fusion of three nuclei. That is why it is called triple fusion, and this is called syngamy. So how many times fertilization took place? Twice. One is syngamy, the other is triple fusion. This, after triple fusion, the resultant product we call primary endosperm or primary endosperm cell, and the syngamy, the resultant product is zygote. Now, what will be the fate of zygote and the syngamy? What will be the fate of the zygote and syngamy? That comes under, so we have already discussed about pollination, about fertilization, Now, what will be the fate of this zygote? What will be the fate of this primary endosperm or primary endosperm cell? That comes under post-fertilization event. So, the primary endosperm and the zygote. This zygote will repeatedly divide, and the primary endosperm will, shall I rub this part? And the primary endosperm will also repeatedly divide. But there is a very interesting synchrony. That means which will divide first and whose division will be followed by other. The primary endosperm division will start first. Why? Because it will convert into endosperm The cell of endosperm is fully reserved with food material. This food material will be utilized when the zygote will divide, it will form embryo, and the embryo require a lot of nutrient for its growth, maturity and further division to form a complete mature embryo. This nutrient will be supplied by the endosperm. That is why from primary endosperm, first of all, the endosperm division will take place, followed by the embryo. That is division of zygote. Now, if I draw a very simplified structure of the embryo, once the zygote is formed, after syngamy, then the cell will divide to form a two-cell stage, and then repeatedly it will undergo division. All are mitotic division. In case of the angiosperm, we see by means of mitosis, the zygote will continuously divide. First two-cell stage, the numerous, and likewise it will form a different shape of embryo. Say for example, a simplified structure I am drawing here. This is what we call a globular embryo. Globular embryo. These are the cells. See how repeatedly the cells subdivide to form a numerous cells present in the globular shape of embryo. Based on the shape, it has given the name globular embryo. So try to concentrate. This is what we call as globular embryo based on its shape. After development, repeated division, the embryo so formed globular shape, what we call as globular embryo. Now another type of shape, what you see here, as you see here, very simplified diagram I am drawing, so that you can also easily draw in your exam very easily, without any difficulty. Very simplified diagram, and it will take very less time. Within few minutes you can draw this diagram. This is what we call as heart shape embryo. Heart shape embryo. So this is the diagram of a heart shape embryo. This is the heart shape embryo. Of course, not yet matured completely. It will undergo maturation further. So this is the heart shape embryo. Globular is a heart shape embryo. Heart shape embryo. On course of maturity, this embryo will form a further shape. Let me draw it here. Let me rub this part also. Once it is getting matured, then further this part, what you call suspensor, and this one, this on course of maturity. This is now almost exactly a mature embryo. It's a mature embryo of angiosperm. So how easy way best you can draw the diagram of embryo development, globular to heart shape embryo and a mature embryo. Within no time it will be completed. Just you require to have a very clear concept. These are the cells of embryo. These are the cells, individual cells of embryo. Like this here also. These. So in that way you complete the cellular structure. This part also. Now these two are called cotyledons. Cotyledons. This is the radical part, and this is the plumule. It is this plumule which will give rise to shoot. The plumule will give rise to shoot. Shoot is the future plant, and radical will give rise to root. So if I rub this plumule will give rise to shoot, and this radical give rise to root. In that way, After fertilization from zygote on course of development, the embryo of different types, and on maturity the mature embryo forms. And for this process of maturation, they require profuse amount of food. This food is being supplied by the endosperm. Now, if you take an example, a very beautiful example, all of you have seen the green coconut. When you open up it, cut it, you get full of water. This is what? This is nothing but endosperm. When the coconut is getting mature, water is there along with the white part, what you call kernel, which you prepare very good sweet dishes. What is it? This is also endosperm. Both are endosperm. Then what is there any difference of these two endosperm? Yes. In a coconut, suppose this is the coconut, you are getting a white part here, what you call as kernel, white part, and you are getting water inside. So this water, it is also endosperm called as free nuclear. If I rub this part, okay, here only, free nuclear endosperm. This is called free nuclear endosperm. And this white part, this part, what you use for preparing various sweet dishes, the kernel part, this is called kernel part is called as cellular endosperm. So what is the basic difference? When the after triple fusion, the product so called PEC, primary endosperm cell, otherwise if we say primary endosperm nucleus so formed. Now this primary endosperm nucleus undergo repeated division to form so many hundreds and thousands of nuclei, and the nuclear division is immediately not followed by cell wall formation, cell division. Only nuclear division takes place. That is called as free nuclear division. Free nuclear division. By means of free nuclear division, it is the water or the green coconut which is full of water. This water is formed. So the coconut water is nothing but the free nuclear endosperm. And after certain maturity, then this cell formation starts. That means the endosperm nuclei, which was produced thousands of nuclei by free nuclear division, will undergo now cell formation. So the outer part is the after formation of cell. So this is what we call cellular endosperm. I suppose up to this much is very, very clear to you. Next few parts, seed formation and other, I shall take up in next classes. Till then, thank you and enjoy learn with whatever we have discussed or which we will be discussing in next class also. Learn by heart. Thank you.